Hey everyone, my name is Alyssa Andrew and I'm going to be showing you how to extract strawberry DNA. I'm a senior from Oregon State University and I'm studying biology with the pre-veterinary option and a minor in chemistry. I graduate next month, but I'll be back in fall to get my master's in water resources science. I'm obviously a huge nerd and this strawberry DNA extraction lab has been one of my favorites that I've done while I'm in school, so I thought it'd be a perfect one to show you guys. It's a great quarantine activity that shows how fun science can be. It's really fast and easy, and you probably have most of the ingredients and materials at home already. So I'll start off by giving you a little bit of the science background to it, like how it works, why we extract DNA, and then I'll show you a step-by-step video that you can follow along at home to extract your own strawberry DNA. Good luck, and I hope y'all enjoy it as much as I do. Thanks.
To start off, what exactly is DNA? DNA is short for deoxyribonucleic acid. Our DNA is what makes us, us. Think of it like a recipe book, where DNA contains the ingredients and directions needed to create the different proteins and traits that make every plant and animal unique. Proteins are what do most of the work in cells and are required for the structure and function of the body. Without our DNA providing the recipe, proteins would not be created properly and we would not be able to function. So, the purpose of this experiment is to extract or remove and isolate the DNA from a strawberry. But what are some of the common reasons for extracting DNA in a broader sense? The first is to study diseases. Isolated DNA can also help us develop diagnostics and drugs to treat these diseases. DNA extraction is useful in forensic science. You can match DNA from a crime scene to the DNA database to catch criminals. Sequencing genomes helps researchers know what makes some species different from others and can help them figure out which species are closely related. DNA extraction can also be used to detect bacteria and viruses, helping with public health. The picture on the right is of strawberry DNA, like you will see later on in this video. The steps followed in this experiment are similar but somewhat simpler to what is performed in labs to extract DNA. The extraction mixture you create is very important for this process, and each ingredient serves a special purpose. The dish soap and the crushing of the strawberry helps to dissolve the cell membrane, which is what holds everything inside of the cell. The salt helps to release the DNA after the cell membrane dissolves. The chilled isopropyl alcohol keeps DNA from dissolving into the mixture. This is because DNA is insoluble in isopropyl alcohol. If all fruits have DNA, why do we specifically use strawberries? There are two main reasons for this. The first is that strawberries tend to have more DNA than other fruits. The second is that strawberries are octoploid, meaning that they have 8 copies of each type of DNA chromosome. Chromosomes are what contains the DNA. In contrast, humans are diploid, meaning we only have 2 copies of our chromosomes. Because of these unique properties of strawberries, their DNA is easy to extract and see, making them perfect for this experiment. Now that we have a better understanding of the science behind this experiment, we can talk about what materials we need. The first is probably the only ingredient that you might not have on hand. You will need 70% isopropyl alcohol, which is also known as rubbing alcohol. I've included a picture of the bottle, and most drugstores usually carry this. However, with an increase in cleaning and disinfecting associated with COVID, it may be difficult to find this, so you might have to try a few different stores. Luckily, my friend had some left over from the last time we did this activity, so that's where I got mine from. You will also need 6 tablespoons of water in a small glass container that can hold about half a cup. If you don't have glass containers, you can use any container of a similar size. Glass is recommended because it makes it easier to see the entire process. You will also need a medium glass container that can hold at least 1 cup. Next, you need 2 teaspoons of Dawn dish soap. I tried this experiment earlier using Ajax dish soap and wasn't able to successfully extract the DNA, so I highly suggest using Dawn. You will also need a 1/4 teaspoon of salt, 1 strawberry, a plastic Ziploc bag, a coffee filter or strainer, and tweezers. Now that you have all of your materials, it's time to extract your own strawberry DNA.
You're going to put your isopropyl alcohol in the freezer to let it chill while you're doing the rest of the experiment. So once you have your isopropyl alcohol in the freezer, you're going to want to fill a medium container with 6 tablespoons of water, preferably glass, but whatever you have at home is fine.
Next, you're gonna add 2 teaspoons of Dawn dish soap. 1, 2. You can leave your spoon in there so you can stir it up later.
Next, add a 1/4 teaspoon of salt.
And then you stir it all together until the salt dissolves so you can't see any more of the little granules. Just give it a nice little stir. It's gonna bubble up a bit, a little bit more. Okay, that's good. Now that you have your extraction mixture, you're gonna take a Ziploc baggie and a strawberry with the top cut off. You open the bag, put the strawberry in, pour your extraction mixture in the bag, and then seal the bag and get as much air out of it as you can. You're gonna wanna make sure it's as sealed as you can get it. If you wanna double baggie it, you can. And then you're gonna mash up the strawberry and try to get as much of the chunks out as you can. You want it as squishy as possible. Okay, so that should be mashed up enough. Let's see if you can see a little bit better. We got most of the chunks out. It's kind of hard to tell with the bubbles. So your next step is to put a coffee filter in the medium container. I like to rubber band mine down to keep it from slipping while you're working. Once you get the rubber band on, you can push it down a little bit, and then you're going to take your strawberry and extraction mixture and you're going to pour it into the filter. You might need to let it strain a little bit before you put the rest of it in. It's totally normal. And you're going to want to get as much of this mixture into the coffee filter as possible. You can see it draining a little bit. So once you have it all in, you're going to want to try and get as much of that pulp or keep the pulp inside the coffee filter, but you want as much liquid out as you can. So you're going to want to squeeze the bag, but do it pretty gently because the coffee filter is pretty fragile. You're just going to gently squeeze out as much as you can. But again, make sure you don't get any chunks into the mixture. Alright, so now that we have it all in the speaker, you're going to want to pour it into a smaller glass container. This makes it easier for the extraction process. Now take your isopropyl alcohol out of the freezer. Now you're going to add 1 teaspoon of the chilled isopropyl alcohol. You just slowly pour it in. And now don't mix it, you're just going to want to let it sit there for a little bit. And you can start to see little white clumps forming, and that's going to be your DNA. You should start to see another layer forming on top. Now you're going to take your tweezers and you're going to just stir it around a little bit. Try not to mix it, but you're just trying to get the DNA to all clump together so you can pick it up in one piece. Here you go. Those stringy bits are your strawberry DNA.